Okay, we complete our discussion on protein structures today, and just to recap on what we've done so far, initially we studied about the different amino acid structures and their properties. We considered the hydrophobicity as the most important property of amino acids and constructed hydropathy plots to determine which regions of the protein amino acid sequence are actually hydrophobic in nature, which would occupy the core of the protein. We then went on to do the protein hierarchy, meaning the different structural properties of proteins from the amino acid sequence to the final quaternary structure which some proteins adopt. Then we consider the secondary structural elements, mainly the alpha helices and the beta sheets, and their different properties as to how they interact and what are their specific features, be it the backbone or the hydrogen bonding properties. Then we constructed a helical wheel to figure out which regions of the alpha helix would be inside or facing the surface of the protein. And then finally we went to certain properties of proteins and predictions of secondary structures. Now that we know something about proteins, we have to consider now the different classes of proteins that are formed based on their structure and solubility. Some of these we have gone over initially. For example, if we look at myoglobin, it is a globular protein. So it is more or less shaped like a globule, which is the case with most proteins. We understand now that on the surface we are going to have hydrophilic residues, and within the core we are going to have hydrophobic residues which tend to remain away from the solvent as far as possible. The structure that you see here is the heme, which we will be studying later on when we consider myoglobin and hemoglobin specifically. Now, if you look on the right hand side, we have the membrane proteins. The membrane proteins, as we studied last time, are embedded in the phospholipid bilayer that forms the cell membrane, and we have these helices that are transmembrane helices, and we also did hydropathy plots to figure out how we can actually determine which of these helices or where they are going to span the membrane. And we also learned that on the surface of these helices, it is likely that we are going to have hydrophobic amino acid residues so that they can interact with the hydrophobic tails of these lipids. There is another class of proteins that are fibrous proteins. From the name, you can figure out what the construction of it is going to be. They are more or less fibers, okay, and they are adopted to give strength to the structure of the protein. So what we have in fibrous proteins, as you can see here, if you have a single alpha helix, you can have a coiled coil of two helices, or you could have what is called a triple helix, as you have in this case. So the interactions here would result in what is called a fiber, and you recognize that these long fibers or large sheets that are actually formed are parts of structures that require mechanical strength. For example, alpha keratin and collagen are two such examples of fibrous proteins, and because of their strength or because of their structure, you find them, for example, in hair or in muscles. Okay, because they are fibrous in nature and they form this long parallel structure along a single axis, they give you this longitudinal structure and they give a mechanically strong nature to this. For example, alpha keratins, they are found in hair, okay, and their sequence consists of long alpha helical rod segments, as I showed you in the previous slide. Beta keratins, the ones that we see here, they form, as you can see, of beta sheets. Okay, they are found in silk, which is also a fiber, but over here, if you look carefully, you can see alanine and glycine repeat sequences, as they are called. So you have alternating glycine and alanine. The reason being, now that you know the structures of these amino acids, they are small in size and they can be accommodated in the beta sheet structure of beta keratins. Okay, so the alpha keratins are the ones that form the alpha helices, beta keratins are the ones that form these beta sheets that consists of a gly-ala repeat sequence. And in the alpha keratins that are found in hair, what do we see? We see longitudinal parallel alpha helices that give mechanical strength to the fibers that are formed. Okay. In consideration of globular proteins, usually they are classified according to the type and the arrangement of the secondary structure. So what you find is you will probably see antiparallel alpha helix proteins that consist of only alpha helices. You might find beta sheet proteins. You might find alpha plus beta or alpha by beta. There are different types of nomenclatures depending on how the protein actually looks. Okay, so in this case you would have a largely alpha helical protein. You could have, for example, when you're looking at a globular protein, what you're looking at essentially is the globular structure of the protein. Now, if you look at this case, these are all globins. Okay, they are all globulin proteins. This one is from plant, the middle one is from the insect, and the last one is from a mammal. What do you notice here? You notice that each of them have very similar structure. The reason being that they are going to perform the same function. So whether it is from a plant or an insect or a mammal, because these globins perform similar functions, they have similar structures. Okay. Now what we're going to study today is what keeps this structure together, because we now know that the only types of covalent bonds that you have in proteins are the linkages between the amino acids that give it the peptide bonds, and what are the other ones? The disulfide linkages that you see that are covalently linked together, which can do what? They can bring different parts of the chain close by. Okay. And we also know of the alpha helices and the beta sheets that also bring parts of the structure together. And what are they connected by? Not covalent bonds but hydrogen bonds. Okay, so what are the forces that are going to drive protein folding? Because now we don't have our polypeptide chain anymore, we have a folded protein structure. Okay, and what we need to know is how this protein folded structure is formed. Now, there are certain terminologies here that I am going to go through. One is called hydrophobic collapse. Now, what we mean by this is that you know that the core of the protein is hydrophobic in nature. So it may so happen that initially when the polypeptide chain is formed, there is a hydrophobic collapse, meaning that there is a hydrophobic core formed first, and the rest of the protein chain then will gradually form alpha helices or beta sheets or interconnectors such as loops or turns to bring about the three-dimensional structure. So initially you are going to have a collapse. The polar surface is going to interact with the solvent, and we are going to have gradual formation of the alpha helices. There may be another case where the alpha helices are formed first. So there would be local interactions first, followed by gradual collapsing of the overall structure. What you're looking for is minimum volume, where you don't have very large cavities, because that would open up the protein. The disulfide bond formation you recognize will stabilize the protein. Why? Because it will bring different parts of the polypeptide chain close to one another. Then you have hydrogen bonds, which we studied in detail in case of alpha helices and beta sheets, which will bring different parts of the protein structure together. Again, we have polar and electrostatic interactions, which we're also going to study and consider how we actually form these ionic interactions or other polar type hydrogen bonding between amino acid residues. Okay. Now, what we have now is we have what is called an unfolded form, that was the polypeptide chain, and we have a native folded form of the polypeptide chain. Now, if we look at the hydrogen bond once more and see how it stabilizes the interactions, for example, in alpha helices and beta sheets, you now recognize that we do have a lot of hydrogen bonds here, and what happens is the accumulation of these hydrogen bonds renders a large amount of stability to the protein. Okay, because it's not just one hydrogen bond that you have a series of hydrogen bonds both in alpha helices as well as in beta sheets. And you also recognize here that in an anti-parallel case, the hydrogen bonding is between similar amino acid residues, and what happens in the parallel case? They are between different amino acid residues. Okay, that's very clear here, and we also did this in the last class. So, what are these structure stabilizing interactions? The covalent, we're not talking about the peptide bond anymore because we already have the amino acid sequence. Okay, so the amino acid sequence means that the amino acids are linked together to form the polypeptide chain already. But now we also know that we have these disulfide bonds that actually form from the link of two cysteine amino acid residues in forming a cysteine bond where we have this SS linkage. Now these can be very far apart in the structure and bring the polypeptide chain close to one another. So we would have a structure stabilizing interaction in the form of a covalent bond in the form of disulfide bonds of cysteine residues. What are the other cases? The van der Waals forces that are weak electrical interactions. They are transient in nature. We have hydrophobic. What are these? These are clustering of non-polar groups together, like we form the hydrophobic core of the protein. Then we have hydrogen bonding. What do we have in that case? We have a donor atom that has the hydrogen attached to it, and we have an acceptor atom. And what happens in this case? We have the hydrogen atom that is sort of partially shared because of the acceptor atom, and the donor atom having partially delta negative charges and the hydrogen being delta positive. Okay, so now what are all these interactions? All these interactions are non-covalent in nature. There is no covalent linkage. But what happens is, when you add these all up, you have a substantial amount of energetic favorability for the folded conformation. Okay, we're going to study that in a bit more detail as we go along. Now what do we have here? This is called what we mentioned about the hydrophobic collapse or a hydrophobic effect, so to speak. When we have a non-polar solute, there is no possibility of hydrogen bonding. Why? Because we do not have any polar group or any group that can act as a donor. What do we have to have? We have to have a donor and an acceptor. But in the case of a polar solute, what do we notice? In case of a polar solute, we see that there is the possibility of hydrogen bonding. So when we have, so what would we expect on the surface of the protein? All these polar residues that can interact with the solvent would preferentially be on the surface of the protein. However, the non-polar solutes that have no such interaction with the solvent around it would tend to be in the central part of the protein, or rather what we call the core of the protein, the hydrophobic core. Now, this is an example of a structure stabilizing interaction. What do we have here? We have an ion pair. What is an ion pair? An ion pair would link a lysine and an aspartic acid together, because you have to remember that it is not always possible for all the lysines or all the aspartic acids or all the basic and acidic charged amino acid residues to be on the surface. The reason being that it may so happen that it follows a large hydrophobic region. So that would tend to be in the core. Then the covalent bond is not going to break, but the lysine is there. So what it's going to have is what is called an ionic interaction, or what is called an ion pair formation with a charge of an opposite kind in the protein chain itself. So that is going to affect the folding, because the lysine is going to be looking for an acidic amino acid residue close by so that it can form this ion pair. Okay, so what is that going to do? That is going to change the folding in a sense. Okay, it is going to affect the folding. In, say, if you had all of these hydrophobic in nature, then the nature of the folding would definitely have been slightly different than what you see here. Okay, what do we observe here? We observe a lysine, that is a basic amino acid residue, have an ionic interaction, an ion pair formation with an aspartic acid residue. Okay, what do we see here? This is a case where you can have hydrogen bonding. Okay, we do not have ion pair formation here, but what do we have? We have a donor. Who is donating? Serine is donating, and we have an acceptor, that is the carbonyl oxygen of the amide group of the asparagine. Okay, so this would be the formation of a hydrogen bond. This would be the formation of an ion pair, and both of these are extremely important in forming your final protein folded structure. So what do we have? Now when we consider energy terms, what are we talking about in energy terms? Let's just look at what energy terms we can have. If we're looking at energy, then we can have the total energy can be covalent, or we can also have a non-covalent component to this. Right? When we're looking at the covalent energy components, because this is contributing to our total energy. Now, under the covalent ones, what do we have? We have bond formation, normal bond formation that is going to contribute. So we have bonds. What else do we have? There is a contribution from angles. There is a contribution from, what other do we have? Torsional angles. Okay. So we have energy contributions from these three components under my covalent set, because these are formed from direct linkages. What about the non-covalent? What are the ones that we listed? We listed a hydrogen bonding, a hydrogen bond. We listed an ionic interaction. And what else do we have? A van der Waals interaction. Okay, so these are the three that we could have in case of a non-covalent formation, and these are the three that we are going to have in case of covalent bond formation. And summing these together, we are going to get the total energy. Now, what do we want this total energy for the protein to be? A minimum, right? We want this to be a minimum so that we have a stable structure. Okay? So, total energy, we have to get a minimum which is going to result in a stable structure. And what is this structure? It is the final native folded form of the protein. Okay? Now, what are these in terms of energetics? If we look at the bonds, how do you represent bonds in just normally for a compound? You represent them as springs, right? So, you have, what do we have in case of bonds? We represent these as springs and they follow Hooke's law. All of you know what Hooke's law is. When you have two masses connected by a spring, they are going to give you some energy following Hooke's law. Okay? What is that? That is this. What is this energy due to? It is due to stretching energy. Okay? This follows Hooke's law. What is this R? The R is the variation that you will get because the bond is not going to stick to one specific distance. It acts like a spring. Okay? Why? Because it is going to form certain interactions. So, it has to act like a spring where it is going to have some favorable interaction that it is going to try to adopt, or it is going to have some unfavorable interaction which is trying to get away from. Okay? So, what we have here is we have our covalent. What do we have in our covalent? We have our bond. Our bond is going to give us an energetic contribution to the total energy in the form of Hooke's law. What do we have for the angles? We have a bending. That is going to contribute to the energy also. These are called energy terms that are added to the total energy that the protein is going to have. Okay? Now you understand that this is going to get extremely complicated if you want to do a computation, because the number of atoms are huge. Okay? But nevertheless, these are the components that form the total energy. We also have the torsional energy. Okay? What you need to know is you need to know what are the contributions. You need to know that the covalent terms come from the stretching, the bending and the torsional. What about the non-covalent? What are they going to come from? We are going to have an ionic interaction, we are going to have van der Waals interaction, and we are going to have a hydrogen bonding. Okay. So a van der Waals interaction, all of you know about the Lennard-Jones potential curve, where we have an attraction due to the negative part of this, a 6-12 potential as this is called, where we have a contribution for attraction and a contribution for repulsion, because you know that these atoms have a repulsive force acting on them when they get too close to each other. Then we have the Coulombic attraction. What is this? This is due to the ionic interaction between the two ion pair formers. What else are we left with? The hydrogen bonding. So this is another feature where we have the hydrogen bonding. Again, we have a 10-12 in this case potential. Now let's look at this once more. What are we talking about here? We are talking about the total energy that a peptide would have, a polypeptide would have, based on its energy terms. Now what are these energy terms? We have energy terms, contributions from the covalent component and contributions from a non-covalent component. Okay? Now the covalent component is comprised of 3 parts. These 3 parts you have to remember from the name itself. Why are these covalent? Because they are directly linked. You have to remember that if you have a spring, it means that the two atoms that we're talking about here are directly linked to one another. If we're talking about an angle bending energy, these are linked to one another. If we're talking about a torsional energy, the atoms are linked to one another. But none of them are linked in a non-covalent formation. I do not have any bonds linking the 2 atoms that have been drawn in the non-covalent set. Okay? So when we're talking about a covalent contribution, we are talking about energy contributions from parts of the protein structure that are linked to one another. One is the bond, the second is the angle, the third is the torsional part. For the non-covalent set, we have the van der Waals, the Coulombic attractions and the hydrogen bonding. Okay. Now, if we look at the interactions and their approximate bond strength in kilojoules per mole, you see that covalent bonds are extremely strong. Ionic bonds are 20 to 40 kilojoules per mole. Hydrogen bonds are approximately 5 to 10 kilojoules per mole. Hydrophobic are 8, van der Waals are approximately 4. Okay? But when we consider a combination of the large number of hydrogen bonds, van der Waals or hydrophobic interactions that are possible, they add up together. Okay? Making it a strong folded structure because of the multitude of interactions that are possible. Okay. Now what I have listed here is what is called an energetic contribution due to suppose, for example, you were to do a calculation to figure out what the total energy of a protein was. Okay? You would use something called a force field. I'm not going into, definitely not going into the details of this because it's beyond this course. But since we consider the different energetic terms, I just want to emphasize here that when you are calculating the total energy, what you want to do is you want to find the summation of all the possible variations from equilibrium bond length for bonds, all the, so what are we looking at now? We are looking at all the possible covalent interactions, the bonds, the angles, the dihedrals. In this case here, this is a very crude one because I have not considered hydrogen bonds here. What do I have here? This is for van der Waals. This is for Coulombic attractions. But I am missing out hydrogen bonds because you understand that when you are doing a computational calculation here, it is going to be extremely difficult to try and calculate all the bonds, because as soon as you form a dipeptide, you recognize how it is going to change or increase the number of bonds, increase the number of angles, and increase the number of dihedrals. Okay? So we have an extremely large number as you go from a single amino acid to a dipeptide to a tripeptide, and if you consider even the smallest protein that would have approximately 30 amino acid residues, that would be extremely difficult to compute, okay? Because you can understand the multitude of interactions that are going to form based on the number of atoms that you are going to have in it. Okay. So what do we have? We have a final native structure. What is this native structure? We have a well-defined 3D structure. It has a specific function, and we remember we spoke about the specific isoelectric point due to the type and number of amino acid residues it has. So what are we going from? We are going from a relatively unfolded structure to a folded structure. Okay? So basically I spoke about Levinthal's paradox last time, where there were a multitude of open conformations that were possible for the polypeptide chain, but it formed this single native structure. Okay? So what is happening now is when it forms the native structure, we have all these interactions that are taking place now. Hydrogen bonds between the alpha helices, hydrogen bonds in the alpha helices, hydrogen bonds between the strands of the beta sheets. Then what are the other interactions that we can have? Disulfide interactions that are going to form the covalent interactions, hydrophobic interactions, van der Waals interactions, and other electrostatic and Coulombic hydrogen bonds and all others between the different amino acids in the protein. Okay? Now we are going to consider the thermodynamics of protein folding. If we consider an initial and final state, what do we have? We have a delta G. What is this delta G? We want to find the free energy change due to folding of the protein. So initially we had an unfolded state, that is G initial. We have now a folded state, so that is G final. So how do we find a delta G folding? We know that is the G final, which is G folded, minus G initial, which is G unfolded. So what do we have to consider? We have to consider, we all know that delta G is what? Delta H minus T delta S. So I have a delta H folding minus T delta S folding. So what is my delta H folding? My delta H folding is again going to be my H final minus my H initial. So H folded minus H unfolded. Now I am doing this at a particular temperature, so the temperature here is constant. What is my delta S? S folded minus S unfolded. Why? Because my unfolded is again my initial state and my final is my folded state. Now we are going to look at the specific terms of these. Now, what happens to my delta, my S unfolded and my S folded? When I consider the entropy, what is the entropy? The entropy is the disorder of the system. Now if I have an unfolded protein and I am now having the protein fold, I am bringing order into the system, right? Now if I bring order into the system, what happens to my S? My delta S is negative. It is not positive anymore. Why? Because I have ordered the disordered polypeptide chain into an ordered folded native structure. So my delta S folding is negative. So what happens to my T delta S? My T delta S therefore is a positive quantity. minus T delta S. Because my delta S folding is negative, minus T delta S is a positive quantity, which is what I have drawn here. So if we look at the energy on the y-axis and on just this axis, the energy axis here, we have an unfolded form here and a folded form here. Now obviously the entropy of an unfolded form is going to be much more than the entropy of a folded form. So my overall minus T delta S is going to be positive. But the enthalpy or the folding interactions that are formed are all favorable. Why? Because a hydrophobic interaction is going to be favorable, a hydrogen bond is going to be favorable, an ionic interaction is going to be favorable. What is not going to be favorable? What is not going to be favorable is if I have a specific hydrophobic interaction trying to make with the solvent. That is going to give an unfavorable situation. But for most of the cases in the folded protein, I am going to have a large number of interactions contribute to the enthalpy. And it is this multitude of interactions which makes this a high negative number, right? So what happens is these more or less compensate one another, and I eventually have a favorable delta G of folding. Okay, so let's go over this once more. We have a delta G of folding that I am considering. What is this delta G of folding? I am going from an unfolded state to a folded state. So I have to consider in the thermodynamic quantities a delta H minus a T delta S of folding. I know that the entropy of a polypeptide chain in the open form is going to be much more than it's going to be in its closed folded form. Okay, so my delta S folding is negative, which makes my minus T delta S a positive quantity. Okay, then I have delta H folding which compensates for this positive ordering of the system because of the large number of favorable interactions that are formed on protein folding, which are all the energy terms that we spoke about in the non-covalent states, right? And we have our delta G of folding, that is a small negative quantity. What does this mean? It means that this is a favorable spontaneous process. Okay, so I am going to go from my unfolded form to the folded form in a spontaneous process. Why? The delta S is not going to make it spontaneous. It is the enthalpy that is going to make it spontaneous. Okay. Okay, we thermodynamics of protein folding. Now what we have here is, if we consider the delta G of folding, we have the initial part which is an unfolded part and a final part which is the folded part. Okay. So when we are considering the delta G, we go from an initial unfolded form to a final folded native structure. So the delta G is G final, that is G folded, minus G initial, that is G unfolded. If we consider now the form of delta H folding minus T delta S folding, we recognize that we can also open these up into the initial and final components. So the delta H folding is going to be H final, that is H folded, minus H initial, that is H unfolded. Similarly for delta S, we can have S folded minus S unfolded. Now, if we consider the entropical considerations, if we go from an unfolded form that is disordered to a folded native conformation, the entropy contribution delta S is going to be a positive quantity, a negative quantity. Why? Because we are ordering the system. A positive quantity is when we have more disorder. A chaotic situation would give you a favorable entropy, but we are bringing order into the system because we are folding the polypeptide chain. So our delta S is negative, so minus T delta S is a positive quantity, which is what we have here. It is positive in its energy contribution. But the delta H folding is a negative quantity due to the different interactions that give favorable energy to the folded conformation. Due to that, we have this positive energy that is due to the entropical considerations compensated by the enthalpy. due to the large number of interactions possible. So the overall delta G folding is a small but negative quantity, meaning that you have favorable spontaneous folding from the unfolded to the folded native protein. Now, if we consider the native state and the denatured state, this is in an equilibrium situation. The beginning of the protein begins at N and it ends at C. So we have a disordered orientation here, that is also called an unfolded state or a denatured state, and we have a native state in this case. So if we have a delta G associated with it, we are going to have an equilibrium associated with it. So we have a K, that is minus RT ln K, the K being the equilibrium between the denatured state and the native state. So the stability of the native state is defined as the difference in free energy between the native and the denatured states. So what do we have in the native state? We have a compact structure, intramolecular non-bonded interactions, and the entropy significantly decreased because of the well-ordered conformation. For the denatured state, what are the features that we have? We have a non-compact structure, we have intermolecular non-bonded interactions, we have a large delta S due to the large number of different conformations. All of these contribute to the delta G of the system, the delta G of folding, which is going to be a small but negative quantity because we are going to have spontaneous folding from the denatured state to the native state. Now, if we consider the different interactions that we had, what were the different interactions that we had? We had hydrogen bonding, we had electrostatic interactions, we had van der Waals interactions, hydrophobic type interactions. Now, I can denature the protein if I add certain denaturants to it or I increase the temperature. Now, we have to consider that in the process of denaturing, I am disrupting the actual interactions that were holding the protein folded together. Okay? So, if I look at all the different conformations that I had, so if I had a protein helix structure and I had some sheets, what do I have? In this folded structure, as I add a denaturant, say I add urea, urea disrupts the hydrogen bonding. Now, if I disrupt the hydrogen bonding of an alpha helix, what is it eventually going to do? It is going to open up the helix, right? Because the hydrogen bonds now that were present here would preferably form with urea. So urea sort of gets into the protein structure, disrupts the interactions that were holding the folded or the protein polypeptide chain together, right? Now, what happens if I heat it? Again, if I heat this, for example, you know that if you heat albumin like egg, what happens? You form a solid mass. Basically, what are you doing? You are denaturing the protein. Denaturing the protein. So, if you have a solid globular albumin structure, when you heat it up, what are you doing? You are decreasing the solubility, you are aggregating it. Why? Because you are opening the polypeptide chain up. As you open the polypeptide chain, what happens? You have this mass formed. And so, each of these structures that are supposed to look identical are opening up into a denatured form, forming a solid mass of aggregate, and you have now a denatured protein. This is a denaturation due to temperature. Okay? So, what we can have is, if we go back here, essentially what we are doing in protein denaturation is we are disrupting all the interactions that were possible. What were these interactions? Hydrogen bonding, van der Waals interactions, and ionic interactions. What can we denature proteins by? By increasing the temperature. We can also add different denaturants. For example, acid. What is acid going to do? How is acid going to change the components, or how is it going to disrupt the interactions? Or we could add urea. or guanidinium hydrochloride. That's guanidinium hydrochloride. Okay, so what can we do? What happens if I add acid? It means I am adding H plus. What happens when I add H plus? For example, all the groups, acidic groups, for example, what comprises these groups? Aspartic acid or glutamic acid. What is going to happen to them? They are going to get protonated, right? So if they get protonated, what do I get? This is what is going to happen on addition of acid. Now, if this aspartic acid or glutamic acid happen to be involved in an ionic interaction with, say, lysine, what would then happen? It would disrupt the ionic interaction, right? So if, now what are we saying? We are adding acid to our protein. Now when we add acid to our protein, any group, any acidic group that had the free carboxylic group in its ionized form would get protonated, and if this aspartic glutamic acid that could form an ionic interaction with basic amino acid residue, with basic residues, what would happen to those? They would no longer form. Once I get into this form, it is not possible for the ionic interactions to form. So any folded structure that would have this ionic interaction with an asp and say a lys, what would happen? This would no longer form. So what would that do? That would open my chain up. Okay? So eventually the idea here is that when you have a denatured structure, you are disrupting the energetics. Denaturation usually does not mean that you are disrupting covalent bond formation. Okay? So if you had a disulfide linkage, you would not actually be disrupting that unless you would put in, say, beta mercaptoethanol, or you would have some other reagents that would break the SS linkages. Because that, you have to remember, is a covalent linkage. You are also not breaking the peptide bond. Okay? The polypeptide chain is intact. So what we can do is we can go from a folded form to an unfolded form by denaturing the protein. You can also, what is called, renature the protein in some cases. So what you would have to do is, say you added urea. What would you have to do? You would have to remove the urea if you want to get back here. And in some cases, you can reform the folded protein. So you are going from your native folded structure to an unfolded structure, and you can reform this back if you, so this would be plus urea minus urea. You could form your folded structure back again. So what we learned today was, we learnt about the protein folding, the energetics of protein folding, where we have covalent and non-covalent contributions. The covalent contributions come from bonds, anything that's connected, bonds, angles, dihedrals. Non-covalent, ionic, van der Waals, hydrogen bonds. Those are going to contribute to the energetics of protein folding. Then when we consider the thermodynamics of protein folding, what do we have? We have a delta G associated with the folding. We have a delta H associated with it. We have a delta S associated with it. Okay. So, all of these fall under the purview of protein folding, where we are considering the thermodynamics, the delta G, the delta H and the delta S, and we have an equilibrium consideration here as well. Okay. We will stop here. Thank you. Okay, we start our discussion on enzymes. We've considered what proteins are, we've studied protein structures, the constituents of proteins, that are the amino acids, and their properties. What we're going to start today is our study on enzymes, which will cover about 3 lectures, and we'll be studying the enzyme mechanisms of a few enzymes, which will be basically ribonuclease, lysozyme and a serine protease. As we go along, you'll see what they mean. Now, you've heard about enzymes. You know that you need them in the body for digestive purposes. You need them actually for any process that goes on in the body. Okay, so whether it's your respiratory process or your digestive process or whatever other process is going on, all of them are enzymatic processes. Okay, so there are specific enzymes that are involved that bring about specific reactions, and you'll see how specificity is extremely important in the way enzymes act. Okay, so basically all enzymes are protein catalysts. Okay, they're protein catalysts in that they are catalytic in nature, but they are proteins. Okay, and what we have here, you know about catalysts. What do? They alter the rate of a chemical reaction without undergoing a permanent change in structure. So, this is exactly what the protein is going to do also. It is going to change, we will see how it exactly does that. It is going to change the rate of the chemical reaction, but the protein itself is not going to undergo any change, even though it is going to bring about a drastic change in the reaction. Now, this is a typical picture that all of you have seen, a typical diagram that gives you the free energy of A plus B. What are these? These are the reactants, and your P plus Q in these cases are the products. So, what do you have? You have a delta G of reaction, which is what? It is the G of P plus the G of Q minus the G of A and the G of B, right? It is G final minus G initial. And what is this? This is your transition state, your activation state that has to be, so this activation barrier has to be overcome to get you from the reactants to the products. Okay? Now, if you look at this, there is a specific nomenclature that we have put here, which sometimes is called the dagger sign. So, we have a delta G dagger that tells us that this is the energy of activation for the reactants to go to this transition state. Now, if we have a catalyst, how does this reaction profile change? It changes in such a manner that for the reaction A plus B going to P plus Q, we have now a catalyzed portion of the reaction, a catalyzed reaction in which our activation energy has decreased. In decreasing, what does it do? The reaction is catalyzed. It means that the rate has been enhanced. Okay? A, capital K is for equilibrium always, and small k is always for the rate. So, we have a small k for the uncatalyzed reaction. I have a small k for the catalyzed reaction. How do I determine it? All I have to do is plug in the values here. A, if we consider the pre-exponential factor to be the same. So, how I have to be careful about units here, kilojoules, 8.314, and by default we usually use 298. Fine. What about my catalyzed? A, E minus 46,000. 8.314. Remember to use the right R also, 2 0.98. So, I can find out how the rate has increased for the catalyzed reaction as compared to the uncatalyzed reaction. It turns out that this is approximately 5 into 10 to the 10. So, what am I saying? I am saying that when I reduce this energy of activation, I have reduced this energy of activation, and what have I done? I have increased the rate 10 to the 10-fold basically, right? So, I have a uncatalyzed reaction, a catalyzed reaction, and then I have associated with it the change in the rate due to the reaction being catalyzed. Whether the catalyst happens to be an enzyme or any other catalyst, it is the same thing. So, what do we have here? We have our enzyme plus substrate, and we have our enzyme plus product. Okay. So, what is our catalyst? Our catalyst is the enzyme. If you notice, what do we have in the beginning? In the first slide that I showed you, what was the first slide? We had A plus B going to P plus Q. What do I have here? I have E plus S going to E plus P. So, what is my E? My E is my catalyst that is transforming the substrate to the product, but itself is remaining unchanged. Okay? So, it is not like a normal reaction where you would have A plus B going to P plus Q. It is E plus S going to E plus P. E remains as it is in this reaction. Okay. So, what do enzymes do? There are certain quantities that you have measured. It is your experiment. What do you know? You know the amount of substrate you have added. You know the amount of total enzyme that you have added, but you do not know at what time how much enzyme substrate is actually being formed, right? So, what you know is these are the quantities you know. You know what S is, and you also know what the total enzyme concentration is, right? Now, at any time t, the total enzyme concentration is going to be what? Whatever free enzyme is left plus whatever has formed the enzyme substrate complex, because the enzyme cannot go anywhere else. It either is free or it has formed an enzyme substrate complex, right? So, that is what our ET amounts to. Okay. So, now if I change the expression and instead of the enzyme concentration, I write ET minus ES. I can write this for the free enzyme. Where do I have this quantity in my expression? Here. So, this is where we are going to now substitute this. So, do that. So, if we do that, what do we get? We get K minus 1 plus K2 ES is equal to K1 ET then minus ES that is what I have. So what can I now do? I have another expression for ES here, which I can take over onto the left hand side, right? So if I do that, I will amount on the right hand side to K1 ETS. If I take this ES part over onto the left hand side, what will I land up with on the right hand side? A K1 ETS. And what will I have on the left hand side? Let's just do it. I will have on the left hand side a K minus 1 plus a K2 plus a K1 S all multiplied by ES, and what is this going to be equal to? K1 We continue our discussion on enzymes and enzyme kinetics. What we discussed last time was Michaelis-Menten enzyme kinetics. Now for this we have a particular reaction scheme where we have the enzyme react with the substrate to form what is called an enzyme substrate complex. Now in this enzyme substrate complex we have an equilibrium as to what is called a pre-equilibrium step, which then finally dissociates into the products E plus P. Now